So the lab activity we're doing today is called plant pigment chromatography. What you guys will get is a piece of chromatography paper. It'll be about 13 centimeters by 15, 16 millimeters. And what you're going to do is you're going to place a spinach leaf along the pencil line that you created about 2 centimeters from one end, and you're going to roll a dime over that spinach leaf so that the spinach leaf is in between the dime and the filter paper, and that's going to squeeze out the juices from that spinach leaf. And in those juices from the leaf are going to be the pigments from the chloroplasts. So every time carefully take your dime, put the darker side of the spinach up. We did a test. It definitely appeared that we got more chloroplasts, a darker color, when we have the darker side of the leaf facing up. So we're going to roll the edge of the dime over the spinach right above the pencil line. So when you pull away the spinach leaf, you hopefully will see a little line of green on top of the pencil line. And after you do that, let it dry out a bit. Maybe put it under the lamp that is at the table for a few seconds. Just a little bit of heat from that bulb will help to dry out the fluid from the spinach. And then I want you to do it again. You're going to do it hopefully around 15 times up to 20 times. The more times you roll the penny or the dime over, the darker that line is going to be, and the better result you should have. So you're going to go ahead and do that 15 to 20 times, rolling the dime over the spinach leaf, getting that line nice and dark. Now, when you're done with that, you are going to carefully cut the filter paper or the chromatography paper so that there's a little an arrow on the side of the line. So just go a couple millimeters down from your line of green, and then cut inward so you get a little tip, a little point. It'll look like an arrow. And then when you're ready with that, you're going to place it in the tube of chromatography solvent so that the solvent is in between the point of the paper and the green line. You got to make sure for sure you cannot let that green line go underneath the level of the chromatography solvent. That's not good. Ideally, you want it in between. The level of the chromatography should be in between the tip of the point and the green line. And then you're going to watch carefully until the solvent front, so look carefully, look the part that's getting wet, it's going to travel up to the other end. By the way, make sure you put the cap back on. The cap has got to be on the test tube, or else it's going to dry out, and you might not get the solvent to go very far. But with the tube capped, it should go all the way, and I want you to stop it once the solvent front gets to within 1 or 2 centimeters of the end. When that happens, carefully pull it out quickly using the tweezers and take a pencil and draw a line along that solvent front. And then let it dry a little bit. You are going to look carefully for those 4 different pigments and look for where the middle of the pigment will kind of spread out over a larger area. Look for where the middle of that pigment is and draw a dot there with your pencil. And remember, you should have 5 markings on your paper. 1 marking is the line for where the solvent went, and then the other 4 should be dots where the 4 pigments are. The first pigment, the first one closest to the line should be chlorophyll B. which is that olive green. Then you have chlorophyll A, which is that more blue-green. Then you have xanthophylls, usually pretty close above chlorophyll A, which is a light yellow. Then carotenes will be very close to the solvent front. It'll be basically along that solvent front. So you're going to draw, just put that little pencil mark in the middle. Then you're going to take your ruler and measure how far each of those dots traveled, each of those pigments essentially. And you're going to measure in millimeters, so you're going to have numbers like 55, 42, 62, 78, 100, 91, those kind of numbers. And when you do that, write the number on the back, or on the solvent paper, and then on the back of your lab paper, I want you to draw, recreate, draw an outline and then recreate your paper as best you can, drawing the shape and size of the different solvents. And go ahead and grab some colored pencils and try to recreate those colors, greens and yellows that you're seeing. Maybe a little bit of orange for the carotene. Okay, so day one, that's what we've done. We've created that line of spinach juice, really concentrated. We ran it for about 30 minutes in our chromatography solvent. We got the separation. Then we identified our different pigments. We identified our solvent front. And then we measured how far each solvent went.